Welcome Genetic Teacher channel. Today, we are talking about bacterial cells stains that used to enhance bacterial cells visualization under light microscope by two stains groups: basic stains, composed positively charged ion to negatively charged bacterial cell membrane, such as crystal violet and safranin stains, while acidic stains, composed negatively charged ion, repelled by negatively charged bacterial cell membrane, such as Eosin and Acid Fuchsine stains, as mentioned in next slide. Additionally, this slide show morphological and shape in bacteria, such as Coccus, Coccobacillus, Vibrio, Bacillus, Spirilum, and Spirochete. And this slide present bacterial arrangement, such as clusters, chains, tetrad, Sarcina, and Pair. And this slide view under light microscope difference between viable and death bacterial cells. So, acid-fast stains distinguish acid-fast bacteria from non-acid-fast bacteria, while endospore stains demonstrate bacterial spore structure, whereas capsule stains display presence of capsules surrounding bacterial cells. Meanwhile, flagella stains show presence of bacterial flagella arrangement, as mentioned in next slide. Thanks for attention. Please like and share and subscribe Genetic Teacher videos and channel.